said, the chief. So I'm very happy then to introduce to you Per-Ola Freskgård, the Chief Scientific Officer at BioArctic, to talk about the brain delivery platform for large drug molecules. Per-Ola is an old colleague from AstraZeneca days, but he also spent many years at Roche and worked for large part of his career on these kind of biological delivery vehicles and another kind of fancy protein engineering techniques. So we're looking very much forward to hearing your presentation, Pio. So I will stop share here and hope you will be able to share on your end.
Yes, I will do that. And I will put it in display mode as well. Hope that works. Does it look as it should?
It looks as it should, yes.
Perfect.
Very good, thanks.
Perfect. Thank you, Per, for the introduction, and then great to be here with you this afternoon and discuss brain delivery of large molecules, various types of modalities to the brain for various brain disorders, and also introduce very briefly our brain transporter platform that we have developed at BioArctic the last 5 years, I would say. We are still developing a bit further for accommodate other types of modalities than I will present today. As Per mentioned, this is an area within neuroscience which evolved very rapidly, and the reason for that is that we have made quite good and major progress the last 10 years or so in the field, which I will come back to. So I will start on a quite high level. During the years, people have tried to use various type of delivery methodologies to get medicines or other things into the brain. And here I just mention 4 different ways that people have tried. Of course, local delivery is something that has been used quite extensively, I think. Of course, that is a quite invasive approach. You normally have to drill a hole through the skull, and you sort of inject your drug quite locally, and is mainly for local treatment of local pathology in the brain. Another research has also been quite actively intranasal delivery. There are some restrictions to what you actually can get into the brain when it comes to size and the property of the molecule, and maybe you don't get a very broad and even brain distribution using that route. One delivery path that has been used and actually used also on the market for a few drugs now is intrathecal delivery, where you inject your drug in the lumbar. You do lumbar puncture in the spinal cord, and through the flow of the CSF in the spinal cord up to the CSF that's circulating around the brain in the arachnoid space or in the ventricles, you see some exposure there as well. It's quite invasive, but still for certain type of diseases is absolutely worthwhile using that delivery system and try to treat various brain disorders. And we have some good successes using that delivery approach. Of course, most of us are very keen on using systemic delivery, and that's what we're using in oncology and other very successful drug discovery area. And then you inject that in the bloodstream. But of course, we all know the issue with the blood-brain barrier. It's a huge challenge at the moment, or especially has been the last 30 years or so, when we're now using more and more large molecules to treat different brain disorders. I think when I started in neuroscience maybe 25, 30 years ago, most of our work was actually focused on small molecules. And we did a lot of work making sure that they had the right property to be actually diffused through the blood-brain barrier into the brain parenchyma and have the appropriate exposure for having some kind of clinical effect. But what we have seen the last 20 years or so is the successful implementation of large molecules like antibodies, especially in oncology. And now we're also seeing other drug modalities like antisense oligos, genetic medicines, and even maybe a little bit larger than small molecules like PROTACs and other things that could be also very useful for treating brain disorders. But as you see on the right side on this slide, if you use intravenous or subcutaneous injection of an antibody, for example, is very, very little of the antibody that actually penetrate into the brain. And I will come back to how this exposure or penetration of an sort of standard antibody takes place. But in essence, it's very, very little, even with an antibody that has quite a long half-life in the periphery, we still see very, very low exposure. And I think one of the reasons antibodies still have been successful, at least now in Alzheimer's disease, where we have 2 approved drugs for treating as a disease-modifying molecules, is that they circulate in quite extensively in the periphery, and over time they penetrate into the brain parenchyma, and that's why we see, even with standard antibody, a clinical benefit to some degree, I would say. So if we could use the blood-brain barrier, it's quite certain that that would be the perfect delivery system. There are a couple of points here which are very, very important. First of all, the brain is the most perfused organ in the body. The BBB, or the capillary network, is estimated to be around 600 kilometers long in the human brain and have a huge surface area of these small capillaries. They are quite small in diameters, and it's also calculated that basically each neurons have its own capillary. So are you able to transport or cross the blood-brain barrier is quite close to a neuron or glia cells in the brain. But maybe also very importantly, and a little bit unappreciated in the field, is actually this is a very scalable process across the species. Because if you scale from a mouse brain into a monkey, that's we normally do in preclinical studies, and into humans, this capillary network is scaling with the size of the brain. So what we actually see when we develop these platforms, they're using active transport across the blood-brain barrier, we see actually a bigger effect using an active transport in a larger brain compared to a smaller brain like a mouse, for example. And the reason for that is that the standard antibody penetrate into a mouse brain more readily than into a human brain because of the size. And I will come back to that in a few slides. So as I mentioned, one of the most successful approach at the moment, I would say, is to use receptor-mediated transport. And there, in this case, is an antibody as a cargo, as we call it, when we transport things. You engineer that antibody to engage with a BBB receptor. So you're basically hijacking the receptor that normally transport other things that the brain needs in an active way. It's called receptor-mediated transport. And then you engage with the receptor and follow that receptor across the blood-brain barrier. And these receptors are typically expressed on the endothelial cells. I don't show the parasites and the end feeds of the astrocytes just to simplify the cartoon. And then when it crosses the endothelial cells, it's released in the parenchyma space in the brain, and then the antibody in this case can reach its target more efficiently compared to a passive diffusion into the brain. There is a potential BBB receptor that has been investigated over the years. The one we have developed the most, I would say, is the transferrin receptor. The validation on that is quite extensive, based on both preclinical data and clinical data. People have also used insulin receptor. Another receptor that is CD98 heavy chain that also been investigated. And there is actually one receptor which is used that are in clinic in phase 1, using the insulin-like growth factor 1 receptor that is used for transporting an antibody against alpha-synuclein for treating Parkinson. A little bit on the development of the TFR development system over the years. This is quite a long time when people realized that the transferrin receptor was identified at the blood-brain barrier, was sort of in the 80s and 90s. It's very, very highly expressed, and it's very likely the main function of the transferrin receptor is to transport iron into the brain using the endogenous ligand transferrin. And then people realized if you use standard antibody against the transferrin receptor, you can interact with the blood-brain barrier in vivo. It was the evidence for transport into parenchyma was maybe that strong at the moment, but at least we saw engagement and quite extensive uptake in the capillary network using antibodies. In this case, the antibody was sort of a standard antibody using the both Fabs binding to the transferrin receptor. And then later on, that sort of setup was used to attach cargos. It could be parts of an antibody or enzymes and try to deliver that into the brain. Then, 10, 15 years ago, engineering of different construct was done. One bispecific molecule was tested by Genentech, for instance, where one of the Fab arm was used for binding to the transferrin receptor, and the other arm was used for targeting the target into the brain. There we saw some improvement in brain exposure, and the AUC Cmax profile could also be very nicely modified. Then the monovalent binding construct that was developed at Roche at the moment, which I was involved in, where the binding module was attached at the back of the FC. And the monovalent binding mode was quite important, as we realized. And we also by using that construct, we maintain the bivalent form of the antibody. So both the Fab arms in the antibody could be used for targeting the target. And then later on, another engineering was made by engineering in the transferrin binding site actually into the FC part itself. And that is also now used quite extensively in clinic by the company Denali. And then a lot of companies now are using the monovalent single chain FV format for delivering antibody and other modalities into the brain. And then I think the reason that we have evolved this now into a more sophisticated construct is that we have learned a lot about the intracellular sorting and trafficking of the antibody or the transferrin receptor at the blood-brain barrier. Another important thing which I will come into briefly is also the engagement with the receptor is very important. I already mentioned monovalent binding. The epitope is actually quite important. The affinity, we know it's important because there we can tailor the AUC profile and the Cmax profile, both in the periphery and in the brain, and also how we position the cargo when it binds to the transferrin receptor in the periphery is also very important, especially for antibody with full effector function. I will also mention that the format that was developed at Roche around 2014 actually showed very nice clinical proof of concept in AD. Is the molecule which is called trontinemab that is now taking forward into phase 3. Okay, back then to our brain transporter, which we call the BT platform. I already discussed the left part of the slide that we're using active transport. I should already mention that I think it's quite clear now that the standard antibody is actually not penetrating the blood-brain barrier. It's actually have another entry point getting access to the parenchyma space, which I have maybe on the next slide. But there's a lot of opportunity using these type of platforms to improve the property of an antibody for a certain disease. Of course, you will boost the brain exposure because you have an active receptor-mediated transport. It's a very fast process compared to a passive diffusion of an antibody. We're talking about hours and days instead of weeks and months compared to a standard antibody. We also see a complete targeting, a very broad and deep brain distribution, which is very, very important for certain brain disorders, obviously. And together, we hope to see improvement in clinical effect because we boast and it's faster and broader brain exposure. This technology also allowed to use lower volume because we're using less of the drug. So convenience will also be something we can improve for the patient. We will use lower dose that also could be important for safety in the periphery, for example. And manufacturing lower cost of goods will also be part of the profile of these molecules. We are now developing the brain transporter platform in 3 different parts. So we have one part that we call BTA. There we have developed the brain transporter for transporting antibody. This is fully implemented now at BioArctic, and also we have active collaboration with that system with a couple of partners. We also have a very nice program where we attach enzymes to our technology. We have a lead program in Gaucher's disease, and we are getting closer and closer to do an IND investment for that program. And finally, what we also have invested heavily in now the last year or so, I would say, making sure that we can transport what we call small modalities. And that could be genetic medicines like antisense oligos and sRNAs and maybe proteins and peptides and so on. What I will talk about today is only focusing on the BTA system where we're using antibody as cargos for this technology. I think this is a quite nice slide because it really illustrates how powerful this technology is. So in this case, we have injected an antibody, standard antibody, into a mouse, and then we have used light sheet fluorescent microscopy. It's a sort of a technology where you can scan the whole brain after you have treated the animal and reconstruct the whole brain, and then localize where the antibody actually sits in the brain. And this is data 72 hours after the dose is 10 mg per kg. And I just want to point out what you see in green is where the antibody is. And it's really striking to see that most of the antibody are actually in the corpus plexus, in the lateral ventricle, which sort of speaks to how the antibody from the blood compartment are filtrated through the corpus plexus into the ventricle system and CSF. And from that, the antibodies penetrate through the perivascular space deeper and deeper into the brain over time. And this is, as I mentioned before, this is quite a slow process, and it takes multiple injection and treatment over time to get the exposure that you need. If we look at the same antibody and we have attached that to our BT system, it's very, very different here. The same conditions, the same detection conditions. The only difference is that we have attached our BT module that you see in sort of the green part under the antibody structure. And you can see in this animal model that also expressed or have a lot of amyloid pathology when it comes to plaques. And we see full target engagement, very broad and nice brain distribution in all relevant brain regions. So the same antibody, same conditions. The only thing we do is we're using our brain transporter binding modules that binds in a very unique way, I would say, to the transferrin receptor. And this is the difference you see between a standard antibody and a BT antibody. Very briefly, I just mentioned for a standard antibody, if we inject that in the bloodstream, it has to be filtrated in the corpus plexus. It enters the CSF in the ventricles, and as I mentioned, through the perivascular space or and slow diffusion into the parenchyma. So over time, you will see some target engagement of the multiple injection. For a BT system, is very, very different. You inject it into the blood and you circumvent that you have to go through all these compartments by crossing the blood-brain barrier through the capillaries, and then you access the parenchyma very broadly and fast. So it's a very, very different compared to a standard antibody. This is some preclinical data in non-human primates, again comparing a standard antibody compared to an antibody where we have attached the binding module to the transferrin receptor. This is actually 24 hours after the injection, again showing how fast this process is. So here we compare and measure the standard antibody with the same detection system with and without the BT system. And for instance, in the striatum is up to 70-fold higher exposure for the BT mAb measured in the same way compared to the same antibody. So it's a very, very powerful technology to deliver large molecules using active transport across the blood-brain barrier. This is a little bit of how we have designed this. So what you see here is the transferrin receptor ectodomain. You have the apical domain in green. You have the helical domain in the middle, which is dimerizing the receptor. You have the protease-like domain in orange, and you have the endogenous ligand transferrin in gray. So our binding module, I just mentioned, we just call it BAT007. It sits on the side, and that's quite different compared to what other people have developed so far, I would say. So that gave us an opportunity to avoid to expose the antibody, what we call in the higher area, because we're not binding to the apical domain. So what we do, we attach our antibody to positioning it very close to the cell membrane. And we have this structure of the binding module, and therefore we could use the N-terminal part of the VL domain of the single chain domain. And then if we position the antibody in the lower part, not exposed towards the blood. So this is when it sits on the transferrin receptor on the blood side, if you like, or other cells that expressing transferrin receptor, like reticulocytes, for example. We think, based on the data we have, that we hide the antibody from the immune system, which is very important when it comes to the safety aspect of this technology. And then the effector function is not accessible to trigger any immune response. And the reason we do that is that we would like to have a sort of an immune silent position of the antibody when it binds to the transferrin receptor in the blood, to prevent immune cell activation or other cells to be activated and lead to safety issue potentially. But when it then is actively transported across the blood-brain barrier, in this case, just used an A beta antibody which binds to plaque, the Fc part is then exposed. And we have a lot of data showing that even though we have the binding modules attached to the Fc part, it triggers microglia activation and plaque reduction, both on AD SNP from AD patients and in vivo in mice models. So this is very important when it comes to the safety aspect of the system. Yeah, so this just shows that if we're using this is a sort of a standard ADCC activity, where you can measure activation of the cells which are engaging with the Fc part of an antibody, basically. And here we compare to a standard antibody attached to another binding module that binds to in the higher area compared to our antibody, which bind to the lower area. And there we don't see any signs of ADCC activation compared to the same antibody, but another binding module that binds to a different site and position the antibody very differently. So the positioning and the epitope of the transferrin receptor is very important when it comes to this part of the technology. This just shows that this is actually what we see in animals, sort of a very key test to see that if you have issues with immune cell activation or if the effector function cause any safety issues, is to look at reticulocyte reduction. Reticulocyte is the precursor cells of your erythrocytes, and they express a lot of transferrin receptor. So if you bind to the reticulocyte through the transferrin receptor and have an exposed antibody with effector function, you see massive reticulocyte reduction. And as you can see here, both for reticulocytes and also hemoglobin in this case, we don't see any effect at all. And this is what you see if you're using antibodies that doesn't mitigate for this. And the same thing on the right side, when we do this in non-human primate, we don't see any reduction in reticulocytes. So our design and how we position the antibody on the transferrin receptor, at least in preclinical model, looks very promising. So where are we now with the BT platform? So I mentioned we have used this for multiple BT mAb construct. We use that, of course, to support internal projects, but also for external collaborations that we have now in place. As I mentioned, we can safely accommodate antibody with full effector function based on preclinical data in rodents and non-human primates. We also have binding modules with different affinity where we can tailor the Cmax and AUC, basically the PK/PD profile in blood and CNS for different types of target and cargos. Of course, we also have shown that increased brain exposure use this technology. We also see enhanced in vivo efficacy in different animal models. So we think we have a highly unique and competitive brain delivery platform. I just mentioned collaboration. a couple of collaborations that we have established the last couple of years is with BMS, where they license not only the platform but also an antibody which will be developed further on, hopefully for treating Alzheimer's. Another deal we did last year, and there it was sort of a platform deal that Novartis aiming to use our brain transporting technology for one of the in-house antibody, which is an undisclosed target in neurodegenerative disorders. So we can sort of either license out a full BT antibody or BT cargo candidate, or we can license out just a platform to be used by other companies. This is our portfolio, and as I mentioned, BioArctic was founded and established on the discovery by Lars Lamfeldt, the lecanemab, which is an antibody against protofibrils, A-beta protofibrils, is on the market now, more or less worldwide, I would say. And there we had licensed out that to the Japanese company Eisai. So that gave us a very nicely royalty income at the moment. We also have, as I mentioned, the collaboration with Bristol Myers, and we also have for this more earlier program with Novartis with this undisclosed antibody target. And in-house, we're working with the program in ALS, where we target TDP-43. As I mentioned, we have an enzyme program where we transport the enzyme GCase for Gaucher disease. It's sort of an enzyme deficiency in the lysosome, so lysosomal storage disease. At the moment, standard of care cannot treat the pathology in the brain, so the idea is to use our BT system, not only treating the peripheral pathology, but also the CNS related pathology. And we also recently started a program in Huntington disease using the platform as well. Yeah, so in summary, we have developed this technology. We have in vivo data that we can transport antibodies, as I show you today. We have also in vivo data that we can transport enzymes. We actually now also have in vivo evidence that we can transport small molecules, but there we are quite early in our implementation and just filing patent to protect our approach, which is different, I would say, compared to other competitors. So we're quite careful how we disclose that at the moment. But these are the three parts of the BT system, and we use them dependent on what type of cargo we would like to transport for a certain disease in the CNS. I just want to end here actually, because this is quite important data, and I mentioned already trontinemab, and it's actually now clinical support of using transferrin receptor as a BBB transporter in humans. So what they have shown, and this is sort of the idea, is instead of using a standard antibody where you have to dose quite high and quite a long time to see some plaque reduction based on PET levels, the idea is to use a beta antibody and more rapidly, and maybe very importantly, removing quite extensively and deeply the amyloid pathology in the brain. And that hopefully will give you much longer time having amyloid-free patients when you do clinical trials. And hopefully that could translate into better clinical effect. We're not there yet, but as I mentioned, Roche presented at the latest CTAD meeting that they now starting a couple of phase 3 trial for this trontinemab antibody, and even in early AD they are planning to start a trial as well to see if they can get into early patients and have even a better benefit for these patients. So I don't know exactly how much time I've used. Yeah, pretty okay, I think. I just like to mention there's a lot of people involved in this, of course. I've listed maybe not all, I hope most of the key people are on this slide. And at the moment, BioArctic is growing based on the successes we have with lecanemab, first of all, and also that we're now developing this platform for both internal and external use. So by that, I thank you all, and I'm happy to take questions.
Thank you very much, P.O. And I urge everyone to